Ladies and gentlemen, I'm here today with my friend John Kellum, originally from Pittsburgh, and now I read from Toronto, in this media expert sponsored by Spectral. And it occurs to me that I have to ask John, why are you in Toronto now? Because you moved from the university, you joined the industry. What was the move that you decided to do?
Well, so thank you, Claudio, and it's a pleasure to be here virtually, obviously, with you. I'm still in Pittsburgh, and although I'm working part-time in Toronto, but full-time for Spectral Medical, I still have an appointment at the University of Pittsburgh, and I still do some clinical work at UPMC, but I'm full-time as the CMO of Chief Medical Officer of Spectral Medical. And you know, I have to say, you know, for you and I have been involved in blood purification studies for many, many years, and we've seen products come and go. And I think that one of the things that attracted me to this particular company and this particular project is that I have always felt, and I think you agree with me, that the way to make progress in the field of critical care in general, and in critical care nephrology in particular, in diseases like sepsis and AKI, are to very carefully select patients for the kinds of treatments that we deliver, and not simply try to enroll patients in large pragmatic studies. I mean, pragmatic studies are fine when you're trying to study something that's, you know, that's very pragmatic. If you're comparing an antibiotic with another antibiotic, you're comparing a Band-Aid with another Band-Aid, that's fine. But if you're trying to manipulate the immune system, or you're trying to, you know, carefully modulate the inflammatory response, I mean, this is not something that you can do in a large pragmatic style. So I'm very much attracted to the idea that we can select the right patients and study them, and not try to, you know, capture, you know, everybody with sepsis. The way, you know, we'll see progress in the field is the same way it was done in cancer, right? We didn't try to treat all cancer with one treatment.
So I see this move, honestly, as a perfect strategy, because when collaboration between industry and academic work is absolutely productive and not blocked by bureaucracy or other things, you may really look into potential advancement in science. And the fact that the industry is involved in product development is, of course, part of the game and part of the business. The important thing is the absolutely rigorous application of science, which is not under discussion, of course, when people like you or others are involved. So, having said that, I think that with the endotoxin activity assay measurement, you have today the perfect trigger for action. Somehow, I can do the parallel between biomarkers and AKI mitigation strategies. And in sepsis, endotoxin activity assay and sepsis mitigation strategies, which are, of course, absorption of endotoxin and then following different therapies. So how do you see, from your perspective, this, let's say, tool that we have available today?
Yeah, so thank you for that. I think that's key, right? So you have a patient in the ICU, they have an infection, let's say they have pneumonia, and you bring them into the ICU, they have pneumonia, you intubate them, they develop some hemodynamic instability, and maybe they need some vasopressor, and you look at this, say, is this a case of septic shock, right? Is the shock a result of the infection? Is it a result of endotoxin, or is it the result of other mediators of inflammation, or is it simply the sedation and the positive pressure mechanical ventilation? And of course, we have strategies to sort of sort out, cardiac performance and intravascular volume and whatnot. But we have, very little in the way of tools available for us to sort out the exact cause of vasomotor paralysis. And it turns out that about 50% of patients with vasomotor paralysis due to sepsis have high levels of endotoxin. The other 50% don't. And so if we study patients or treat patients with an anti-endotoxin strategy, and we're including 50% of patients that can't benefit, it's going to be really hard to demonstrate effect. And we're not going to be providing the best care to our patients. And so I think this is a really, as you say, it's a synergy between the diagnostic part of personalized medicine and the treatment part. You need both. And so I think EAA is a good endotoxin activity assay, is a good example of what we can do when we pair a diagnostic with a therapeutic.
What is the half-life of endotoxin?
Good question. So as you know, most of the endotoxin that we actually have in our body isn't present in the bloodstream. It's circulating, right? So even the endotoxin that gets into the bloodstream is taken up by immune effector cells. It's intercellular. It's very hard to measure. And this is the whole reason that endotoxin activity assay was developed in the first place, because measuring endotoxin is using a LAL assay or something is very challenging. And so endotoxin is hiding in different places where it still may be biologically active. And so it doesn't have, I think, a traditional half-life in that respect.
Would you recommend to measure sequentially in a patient who is under suspicious septic condition, to measure sequentially the endotoxin activity assay?
Well, I think that the use of the endotoxin activity assay has been, largely focused on identifying patients early in the course of sepsis, where you're trying to understand the risk of development of septic shock and high mortality, and to identify patients that you need to be more aggressive in terms of caring for. But you're quite right in the sense that the application by clinicians outside of what shall we say, traditional application of these diagnostics can yield a lot more information. And I think that it's sort of up to the clinicians at the bedside to kind of decide how this can be used. I think that certainly the other application for serial measurements of EAA are that patients can be admitted for one thing and develop sepsis down the road. And we're certainly seeing this in the COVID epidemic, right? Where patients have single organ failure, they have, respiratory disease from the COVID, but they're otherwise hemodynamically stable. They don't have other organ system involvement. And then suddenly things change. And the application of diagnostics like EAA can be important in that setting.
Yeah, we have been, doing in some patient measurement every 6 hours. And this was the possibility given to us to start with hemoperfusion, with polymyxin B therapy much earlier compared than in other situations where monitoring was less frequent. Now, another question is, what is, in your opinion, more important, the positive predictive value or the negative predictive value?
No, I think it's, the discovery of two cutoffs really for the endotoxin activity assay. If you're above 0.6, you have very high levels, you have very high risk of death. But if you're above 0.9, you may have so much endotoxin on board that it's not possible to sort of treat patients. And so I think clinically we with the availability of PMX, which of course you have in Italy, both positive and negative predictive value are important, because you don't want to be necessarily exposing patients who can't benefit from therapy. If they're very low endotoxin levels, they're not going to benefit from endotoxin removal, but also positive predictive value, because if a patient has evidence that they have endotoxin, you want to act on that as quickly as possible.
Well, I was always puzzled by this, because in one side, if you are below 0.6, it doesn't mean that in the next 6 hours you may not go above. So that's why I always advocate the use of frequent measurement. On the other hand, when you are above 9, we know that this is an overwhelming concentration of LPS that may not be sufficiently removed in treatment classic with the PMX. However, this may also suggest the possibility to develop different cartridges, bigger maybe, or change more frequently the cartridge, or simply prolong the therapies that we have done for 2 hours or so on. So I think that there is a wide spectrum of studies that we may do, considering that unfortunately the kinetics of these biological markers is not like the kinetics of urea, right?
Yeah, yeah. And I think the other challenge, of course, is that, and you sort of alluded to this in a symposium you and I were chairing also at this meeting, that, RCTs have limits, right? And so we know from the Euphrates trial that 17% of the patients had EAA above 0.9. So then you say, okay, I'm going to enroll patients in a clinical trial, and I only find 17% of the patients who have septic shock have this. And where are you going to find enough patients to randomize into a clinical trial? So there's a limit to what we can do. And I think that finding patients with sufficient organ failure and EAA levels between 0.6 and 0.9 is doable. If you're focused on greater than 0.9, I think in an RCT that would be a big challenge. And so there may be other approaches to take.
And what is your ideal endpoint? Is it a short-term endpoint, or is it a long-term endpoint? I mean, forget what the FDA would like to see, of course, for the moment. Just tell me, because when you have a treatment, my view is that the endpoint should be the effect of that treatment and what the treatment claims to be doing.
Yeah, I think it's interesting. I think that they're both important, obviously. And I think if you look, for example, and you remember the survival curves from your trial with PMX, they were very, very separate for the first 10 days or so. And although statistically significant out to 28 days, they began to come together. And I think that it's important both that the treatment has short-term efficacy, because that's what you're aiming at. But I think it's also important that, at the end of the day, you're sending patients home and at a much greater rate. So I think that, would it be beneficial to have something that worked to preserve organ function and avoid severe clinical consequences of endotoxin, and simply not demonstrate an improvement in mortality? I think, we could definitely discuss that. I think that, if your 28-day mortality were not quite statistically significant, but still, trending in the right way, and you've had benefit in terms of short-term organ failure response, et cetera, I think a lot of people would opt for that therapy. And certainly it's similar to what we're, proving for cancer therapies and things like that. There are things other than mortality which are clearly important.
No, and then of course the opposite is the same when they say the curve of mortality and admission to ICU for COVID is decreasing. It may decrease down to 1 case per year, but if you are that case, you are very much concerned. So this is the question. But the point is endotoxin activity assay and then hemoperfusion with PMX may not be the sole solution for sepsis. What do you think about what we suggested for a long time, to have a sequential extracorporeal therapy so that we can catch the need of the patient in different moments in the, let's say, state in the ICU?
No, I think it makes a lot of sense. And, if you think about endotoxin sort of as the fire and the cytokine response is sort of the smoke, it's true that both the smoke and the fire will kill you, right? So it's you can't just address one and ignore the other. Now, the hope, of course, is that by addressing the fire, the smoke will never fill the room or will dissipate quickly. But there may be circumstances where you need to have both aspects addressed.
Okay, excellent. So I think our next editorial probably should look into this, and we may also include the possibility to go even before the development of high level of endotoxin with the source control and possibly the absorption of other molecular patterns or even bacterial patterns that are important. With this, I think I can thank you very much for this short interview. It has been always a pleasure discussing with you, and I'm really looking forward to next year, possibly in person, further discussing all these issues. For the moment, let's go back to the mainstream of the scientific program, just reminding everyone that this content of the industry symposia are strictly linked to the whole scientific program of the Vicenza course. Thank you very much, John.
Thank you, Claudio.